Welcome, everyone. My name is Haley Cavalier, and I'm with CHE, the Collaborative for Health and Environment. I'm going to start with some quick housekeeping items. So today's webinar is showcasing CUSP, the research cluster to understand the health impacts of micro and nanoplastics. This cluster, funded by the European Commission, consists of 5 distinct projects and research teams, each dedicated to studying a different specific aspect of the health impacts associated with micro and nanoplastics. You can learn more about CUSP at the CUSP website, for which the link will be shared in the chat. And this webinar will focus on highlighting recent findings from 2 of the 5 projects, AURORA and POLYRISK. This webinar is hosted by the EDC Strategies Partnership. The partnership members include the Collaborative for Health and Environment, Healthy Environment and Endocrine Disruptor Strategies, or HEATS, a program of environmental health sciences, Commonweal's Biomonitoring Resource Center, and the Health and Environment Alliance, or HEAL. We'll disable the chat during the presentation, but please type your questions in the Q&A box at any point, and we'll get to as many comments and questions as we can after the presentations. If you'd like to activate closed captions, you can do that by clicking the button at the bottom of your screen. This webinar is being recorded and it's scheduled to last for an hour. If you'd like to download today's slides and the links to the text of the studies mentioned, you can find them on the webinar page and our website at healthandenvironment.org, and we'll put that direct link into the chat in just a moment. So this is such an important and interesting topic given the extent of micro and nanoplastic pollution that we face. So we're very excited to learn from our speakers today. Today's moderator is Janan Jensen, the Executive Director of Health and Environment Alliance, or HEAL. Janan, I'll pass it over to you.
Hello, everybody. It's really great to see so many people online from here in Europe. My name is Janan Jensen, and as Hallie has said, I'm the Director of HEAL. And it's a real pleasure to moderate this webinar today because it's looking at the research being done in the European Union on the health impacts and micro and nanoplastics. And as you've just heard, the CUSP network is composed of 5 collaborating research projects: AURORA, ImpTox, PlastiCheal, PlasticsFatE, and POLYRISK. And the findings from these research projects can really have far-reaching implications for European policies on chemicals, plastic, food, and water. And one of the exciting things is we're in, I think, the 4th year of these projects, and early next year, this CUSP network will actually have its final event and conference in February in Brussels. And there we will be bringing the scientific findings as well as a stakeholder panel to the newly elected European Parliament and the EU institutions to highlight how they can use this to have more precautionary policies. Today, however, in this webinar, we're not going to look at the whole CUSP network, but you can find much more on the resource page. And we will rather deep dive into the work of 2 of the projects from the cluster. One of them is AURORA, and the other one is POLYRISK. HEAL is a partner in the POLYRISK project and works on contributing to the dissemination and policy implications found from the research. So now I'd like to introduce our 2 speakers that we have for today's webinar. Our first speaker is Dr. Hubert Derven from POLYRISK, and he will be presenting research on the exposure to micro and nanoplastics in real-life scenarios and the potential adverse effects on the immune system. For example, in indoor soccer fields, with artificial turf, with rubber granulate infill. He'll also discuss the knowledge gaps related to exposure and human health effects. And Dr. Derven is the departmental director at the Norwegian Institute of Public Health and a European registered toxicologist. At present, his department is really responsible for delivering knowledge-based evaluations on REACH chemicals to the Norwegian Environmental Agency, and he's also involved in several other research projects at EU level, including ExQuimos, POLYRISK, ImpTox, and PARC, looking at human biomonitoring. Our second speaker is Professor Roel Vermeulen from AURORA, and he is presenting research today on how to better understand the MNPs and the effect on pregnancy and development in early life. Professor Vermeulen is a professor of environmental epidemiology and exposure science at the Institute for Risk Assessment Sciences at the Utrecht University. and also at the Julius Center for Health Sciences and Primary Care at the University Medical Center in Utrecht, the Netherlands. Professor Vermon's scientific research focuses on environmental risk factors for cancer, cardiometabolic, and neurological diseases through inter- and transdisciplinary research. He also coordinates the Dutch research program on the exposome, leads an EU project called Expanse as part of the European Human Exposome Network. So as you can see, we have two very amazing experts with us from the scientific field, and we're really grateful that they can be here to share their time and their expertise. So we have someone from the Netherlands and also from Norway. And I'd like to now give the floor over to Dr. Derven to give the first presentation. Thank you.
Thank you, Jiron, and thank you for the opportunity to present some of the results in POLYRISK. This is still ongoing work, so there's still, we have some data, but there's much more to come in the coming years. So POLYRISK is one of the tasks is the human exposure and health effects of micro and nanoplastics. The plastics is a really remarkable material, and the production is really huge. There is a lot of plastics produced in the world, and without any interventions, the production of plastics will be doubled in 2050. But that is something that, for example, the United Nations likes to prevent, because plastics also has a big waste problem. For example, only 10% of the plastics currently is recycled, and the majority of all the plastics that have been produced in the last 50, 60 years is still in use or is discarded. So it ends up in the environment. The plastics is really a remarkable material. It's not only a problem, it has a lot of benefits. You can find it in toys, you can find it in furniture, you can find it in food packaging, and it is really important to avoid microbiological contamination of food. You find it in electronics equipment, you find it in medical devices, which is a really important role in order to make sure that the procedures in a hospital, for example, are really safe for the patient. You find it in textiles. So it's really a material with a lot of applications. But plastics, if ended up in the environment, it can result in these micro and nanoplastics. And this is just an illustration what we are talking about when we are talking about micro and nanoplastics. So originally we have macroplastics, which is quite big. Then it's transformed to mesoplastics and then microplastics. And in this presentation, we will use the everything smaller than 1 millimeter is called microplastics. And nanoplastics is then 1 micrometer. So that is 1 millimeter divided by 1,000. So it's really small. You're really talking about the sizes of viruses if you're really talking about nanoplastics. Sources of micro nanoplastics is breakdown of plastics in the environment. That's the way the majority of the plastics are destroyed in the environment. Sometimes they are really added to products, like in cosmetic products, in detergents, paints, and fertilizers, and plant products, plant protection products. They are on purpose added by the manufacturer. But there is now a REACH registration in place in Europe in order to avoid this intentionally added micro nanoplastics to products. So hopefully the contribution of this source will not be as large in the future. If you look in Europe, then what is the major source of micro and nanoplastics? Paints, and especially if you remove paints from structures, you have painted it and then you want to remove it. That is, according to this study from the European Union, an important contribution to micro and nanoplastics. Tires is really a big source of micro and nanoplastics. Plastics, when they are moved from the factory to places where they are used, it's often in the form of pallets, and these pallets sometimes they are lost during shipments and transport, and they are also a source of micro and nanoplastics. And of course, textiles and detergent capsules is also a major source of micro and nanoplastics. So if we are exposed to micro nanoplastics, probably the majority it will come from ingestion, so that we have oral exposure. So it's taken up from the intestine. Inhalation is probably a source of exposure as well, and often it's mentioned that dermal exposure is also possible. Although then probably you have to have a wound, for example, in your skin, in order to have a major contribution to the internal exposure. But I think the research that has been demonstrated in the last years, I think it's fair to say that there is really evidence that these small micro and nanoplastic can translocate to the blood, so they can pass the epithelial barriers, both in the your oral digestion system and in the lungs, so they end up in the blood. And what happens then in the blood or in the body, that is a little bit more unclear. There is some indications that they are excreted via the bile and feces, or even urine samples. But it's also likely that they stay in the body, for example, in organs like the spleen and the liver and in the immune system, because they have phagocytic activity, and they can probably bioaccumulate these particles. If you talk about possible health effects of micro and nanoplastics, then there is a lot of variables that you need to look at. The size, of course, and especially the micro and especially the nanoplastics are of worry. We have the surface chemistry, the charge, functional groups, protein corona can play a role. Sometimes endotoxins are present at the surface of the micro nanoplastics. The shape is, of course, important, especially fibers. We know this from the asbestos exposure, that we should be afraid of these fibers. Materials, we have a lot of plastics that are in commercial use, PET, polystyrene, polypropylene, PVCs. And then a topic that is getting more and more attention, and I will also come back to that, is these additives. You know, plastics is a polymer, but a lot of substances like plasticizers and flame retardants and pigments are added to these plastics. And also some of these additives are not really added to the plastics, but they are formed during manufacturing. So they are called the non-intentional additives. And that can be quite a significant amount of these additives. So what do we know about micro and nanoplastics in human tissues? Well, there is a lot of publications the last 2 years, and I will only mention some of them. There are much more publications available. This is a publication from Heather Leslie, that is a partner in from the Free University in Amsterdam, and partner in the POLYRISK project. They are responsible for all the micro and nanoplastics determinations in blood. They described in 2022 in this paper in Environmental International that they found polymers in blood as measured by GCMS pyrolysis. There was a study in this year about placentas, human placentas, in which they also found with GC pyrolysis, GCMS, the presence of polymers in human placentas. In all 62 placentas that they investigated, they found these polymers present. A group that did a slightly other approach, because they really looked at with microscopy about the micro and nanoplastic exposures in plaques in the aorta. And they discovered in a number of plaques, indeed, that there are micro and nanoplastics present in the plaques, which is not completely a surprise, but still, it's nice that it has been demonstrated. Little bit more of worry was that they also found a relation between the amount of micro nanoplastics and the increased possibility of people dying because of cardiovascular disease. So that is something, there is a link with an health effect. And then recently, not so long time ago, in May, a study about microplastics in testes, in both dogs and humans, was described. And they found quite a lot of different polymers in human testis, and they also speculate that there is an association with some biological effect, like sperm counts and increase in the weights of organs. So this is only in 4 papers, but there are many more. A lot of organs people discover these micro nanoplastics. So how are these micro nanoplastics measured? It's often that you digest a sample, a biological sample, you ultracentrifuge, and then you have a pellet. And then you can look at a microscope or a scanning electron microscopy and look for, and later on with specific analytical methods confirm that it is indeed a plastic that you are looking at. But the most studies they use this pyrolysis GCMS, which I will describe in the next slide. So in POLYRISK, we are also using scanning electron microscopy together with Raman spectroscopy in order to find out what the chemical nature is of the plastic particle, of the particles that we see. And this is mainly done in air samples because in biological samples this is quite difficult to find these particles in blood or in other organs. But the big problem is contamination. You really should avoid that that you contaminate your samples with plastics. And especially, in every laboratory, if you look around, you see a lot of disposables, pipette tips, and tubes and other things, and they are made from plastics. And you really should avoid that you use these plastic materials during the processing of the samples. Even the samples in which the tubes in which you collect the blood samples is from plastics most of the time. So we had a hard time in finding glass blood sampling tubes, so that we don't introduce these plastics in our samples. And one of the things that you really need to do is if you look at these analytical with these analytical methods, is use a lot of blanks to make sure that during the processing of the samples that there is no contamination. So that is quite critical. This GCMS is then often used. It's a small. The sample is in a small sample cup. That sample cup is placed in this pyrolyzer, and then at high temperatures, 500 to 700 degrees Celsius, in an oxygen-free environment, then the plastic is transformed to small volatile substances, and these are then separated by a standard GC column, and the mass fragments are detected by an MS detector. And this is the method that is used at the Free University in Amsterdam, and Lorenzo Schibetti is the person responsible in the POLYRISK project for doing this pyrolysis GCMS analysis. Then, in POLYRISK we have a very focus on the immune system, because we expect that the immune system are exposed to these particles, and probably immune cells take up these particles in order to remove them from the circulation. So we have a strong focus on the immune system, and especially, for example, cytokine release. We did some literature searches. Igor Snapkov in our department did some work on this. And if you look, for example, at the lung and cells like the A549 cells, there is indeed some data that IL-8 is released from these cells when they are exposed to particles. The same for the gastrointestinal tract. Again, IL-8 is found in cells that represent this organ, and also IL-1B is found. In immune cells, also from our own work, there are even more cytokines that are released, like IL-2, IL-4, IL-6, IL-8, TNF-alpha, VEGF. So this is there is an effect of biological immune cells when they are exposed to particles. And this is work in progress by a postdoc in our department, Neema Negi. She is isolating cells from human blood with a focus on monocytes and dendritic cells. And then we got two types of nanoplastics from Corina Altmann from the BAM Institute in Germany. One is polyethylene with a size of 350 nanometers, and the other one is polypropylene with a size of 180 nanometers. So what NEMA is doing in the lab is isolating these dendritic cells and the monocytes in separate 96 well plates. And then she is treating these cells with these particles. And then, of course, look for self-viability. She's looking with Luminex system to find out what kind of cytokines are produced. And for example, what I presented on the last slide, this IL-8, TNF-alpha, IL-6, and IL-1B, that are cytokines that she found after treating these cells for 48 hours with these polyethylene and polypropylene particles. And then what is ongoing is then not only look at the cytokines, but also look at some markers on the immune cells. And we use then flow cytometry in this particular case. And we see that indeed some of these markers like CD11B, that there is a decreased expression of that marker in monocytes, and that is indicating, but still this is still work in progress. So we need to confirm this with further experiments, that there is an inflammatory response in these cells. So this is nice in vitro work, but what we really like to do in POLYRISK is to study human exposure to micro and nanoplastics. So POLYRISK is an EU project, like already stated, which will end next year. POLYRISK coordinated by Dr. Raymond Pieters from the University of Utrecht in the Netherlands. And one of the work packages in POLYRISK is studying the real life exposure of humans to micro and nanoplastics. So we like to study the external and internal exposure to micro nanoplastics, and also to study biomarkers for effect in blood, urine, and sputum. So we have 5 real life studies planned. One is so-called traffic study, because traffic generates a lot of rubber particles in the air, and we are looking then at different locations with varying amounts of these rubber particles, and we are looking for example, for immune effects and lung effects. We have a study in the textile factory in Romania, because in the textiles, a lot of plastics are used in the production of plastics, and we expected that there is quite an exposure to micro and nanoplastics in these factories. We have a study in a tire refurbishment factory study planned, and then in Norway at our institute we are planning to do 2 studies: a football study and a study with bottled drinking water. And the football study is a study in which we have 36 volunteers, quite young, 17 to 19 years old, from both males and females. And they played football matches on artificial turfs in indoor halls, because in Norway we have outdoor halls with these rubber granulates, but we have also quite a lot of indoor halls because of, in the winter people still like to play football. And there is a lot of snow in Norway. So what we did in this study is we had the same group of 36 volunteers. First, they played a match on a football field, artificial turf with without rubber granulates, and it was crushed olive stones that was used as infill. And then we took biological samples before the match. They played the match 2 times 45 minutes, and then biological samples after the match, and biological samples after 18 hours after the match. And then exactly the same persons played another match on artificial turf with rubber granulates, and again we took the same amount of samples. So we took blood, urine, and saliva, and also we did a lot of air sampling to find out what kind of concentrations of micro nanoplastics were found in the air. So what this is the preliminary result of the studies that we performed. First of all, the bottled drinking water study. We looked at bottled drinking water that are for sale in Norway in different shops. And we found very, very low levels of micro nanoplastics, bigger than 1 micrometer, because that was the amount of plastics that we could identify. So the levels were so low that we thought that it would not make any sense to do the study. So that's good news for the people in Norway drinking this bottled drinking water. In the football halls, we also found quite low levels of rubber, total rubber in air samples, and we are now still waiting for the determination of micro and nanoplastics in air samples. And then we, in the blood of the players, we looked for, we did immune cell profiling with CyTOF, which is a more advanced flow cytometer. And we looked also for cytokine with the O-Link platform, and that's still ongoing, and I will show some preliminary results. And then Lorenzo has a lot of samples now, full blood samples in which we like to measure rubber and plastic levels in the blood samples, but we have not seen the data from that work yet. So what the conclusions that we have at this moment can draw is that we have relatively low levels of rubber detected in the air samples in both halls. The level in the hall with the rubber granulates were slightly higher than in the other hall, but still it was below the level of quantification. We are waiting for the micro and nanoplastic measurements in air samples and in blood samples. We see, however, that there is one metabolites of phenylalanine is slightly higher in people after playing the match on rubber in the rubber granulate infall. So it indicates that there is some exposure to rubber. We see some minor changes in the immunological profile of blood cells with CyTOF, but we see some significant changes in plasma cytokines, and especially in the rubber granulate hull. But still, this is still some preliminary data, so we are still looking further into it. But what we did, and this is work by Igor Snapkov, we looked at we compared the cytokines in the, for example, olive hull before and after the game, and then compare it with the rubber before and after the game. And then we see 11 cytokines that have been changed in both settings, but 7 only in the place after the playing in the hole with the rubber granulates. And we have the same picture if we look for other compounds. So it looks that playing on a field with these synthetic rubber granulates, that there is there is a change in the cytokines in the blood. So then the knowledge and challenges and knowledge gaps, I really think analytical chemical characterization is problematic. There is a high risk of contamination, so always be critical when you read something about micro and nanoplastics in all kinds of tissues. Have they really taken prevention that contamination it can be ruled out? Yeah, I still think that we have not a lot of exposure data to perform a risk assessment. And also the nanoplastics, we lack sensitive analytical methods for nanoplastics. There still the methods need to be improved. Also, the throughput of these analytical methods is quite low. So really biomonitoring of exposure to micro nanoplastics on a routine basis is at present not possible. Also, we need to realize that the particles that are studied in vitro studies in biological systems are often less relevant for human hazard assessment. It's often particles that you can buy commercially, but they are not the particles that we are really exposed to. And I really think that the big unknown is chronic exposure. What are the effects of chronic exposure to micro and nanoplastics? That is really something we should get more data on. And I just want to mention briefly that there is an opportunity. We have a big European project which is called the Partnership for the Assessment of Risk from Chemicals, PARC. And there is an opportunity to perform an OECD type guideline study in, for example, rats, looking at the long-term exposure of micro and nanoplastics. But the problem is finding a representative, well-characterized test item, the nanoplastic. That is a huge challenge. So preferably it should be really nanoplastic, so around 100 nanometer in diameter. It should be available in sufficient amounts in order to do a study like that. And it should be well characterized, and there should be no endotoxins presence on these particles. So if someone has a good idea about where we can get hold of this test item, then please let me know, and we can continue with designing and study on this topic, and filling a major knowledge gap on the chronic toxicity. And then I already indicated that there is more and more people interested in on the chemicals that are added to plastics. This is a report from the PlastiChem project, led by Martin Wagner from the Norwegian Technical University in Trondheim, recently published, and you can find it on the internet. And they listed 16,000 chemicals that can be identified in plastics, which is of course a huge amount. And most of these chemicals are not regulated internationally. It's only 6%. A lot of these chemicals are persistent, bioaccumulative, or toxic. And yeah, we lack hazard data of 10,000 chemicals, and of course we are not exposed to single chemicals. We are exposed to mixtures, so that would also be interesting to study this. And then in the report, a number of priority groups of concern are listed. So yeah, if you're interested in what we as CUSP consortium think about health effects of micro and nanoplastics, I would recommend to read this policy brief that was recently published and which is widely available. Yeah, the conclusion is that micro and nanoplastics and public health, there is a reasonable concern. So it's something that needs further attention. And yeah, one of the reasons is that the MNPs meet the persistent and bioaccumulative criteria in the PPT criteria, which is quite a leading concern in Europe. And of course, MNPs are also mobile in the environment. So I like to thank a number of people in my own group, but also people from the University of Utrecht and the Institute in Romania, and the people doing the analytical work. And there are many more, and some of them are shown on this picture, which was taken 2 months ago when we had a consortium meeting in Norway at our institute. And these are the partners that are collaborating in POLYRISK. And like already stated, we are working together with the other 4 projects in this CUSP cluster, and there is a dedicated website on this for this CUSP cluster activities where you can find much more detail. So thank you for your attention, and I give the word back to Jenon.
Thank you very much, Hubert, for this excellent and very informative presentation. I think we have a lot of information to dive into, and I think particularly interesting will be some of the results from the real-life exposures that you mentioned, particularly the football turf. I think a lot of people are interested in that. And I would like to ask Roel to put up his slides. And I also wanted just to remind people that they can put their questions and answers into the Q&A box for questions at the end. And with that, I will thank you again and give over the floor to Rolf.
Thank you very much. So let's see. So I think you see my slides now and can hear me. Apologies for the slight delay in the presentation. I'm very happy to be here tonight or afternoon or morning, wherever you are, to talk more about the AURORA project, which is one of the 5 projects within CUSP, focusing really on early life health risks, with the idea that we want to develop a roadmap for risk assessment of micro nanoplastics in relationship to early life health risk. I also want to thank Hubert Derven, who actually presented a lot of the background of micro nanoplastics. Just to reiterate, what are we talking when we talk about plastics? Most of these plastics till date come basically from oil or natural gas, where we basically manufacture monomers. We make out of those monomers polymers, which we then basically transform into plastic pellets and then into plastic products, which basically then come to the market. As Hubert already pointed out, when we talk about plastics, we're not talking about polymers only. to give basically the properties to the plastic products, we add a lot of additives, may that be plasticides, flame retardant, stabilizer, antioxidants, and so on, to basically produce the product that we actually want with the characteristics we want. As Hubert pointed out, plastic is not a new exposure, something that has started to increase since the 1950s, when plastic was more and more introduced into common household goods, and basically over time that has increased, as Hubert pointed out, with the amount of production. Indeed, plastics have a lot of characteristics which have been very helpful for modern society, but there's also a large downside, and that is that the enormous increase of the use of plastics has led to a widespread dispersion of plastics or plastic waste, which basically has led to large exposures to ecosystems, basically I think very indicated by the picture on the screen. So in all means, basically, plastic is a large planetary health problem, not only a human health problem, but it's basically a large health problem. And from a sustainability aspect and an ecological aspect, I think there's all reasons to try to reduce the use of plastics in general. However, of course, the large use of plastics and the dispersion of plastics has led also to human exposure. What I show on this slide is a summary that I did basically at the end of last year. So there are many more publications now, but this is basically summarizing the data of about 50 studies that were out at that time. And what you can see here indeed is that these micro nanoplastics are basically found in feces and stool, placenta, meconium, blood, lung, breast milk, semen, testis, and many of the organs. And perhaps we should not be surprised about that finding. If we just think about particles, if we think about air pollution, for instance, we have known for a long time that small particles in the nano size can actually transfer from the lungs into the bloodstream and reach every organ. So I think what we see here is actually a reflection of the technological advancements that we have had over the last few years in actually being able to detect these micro nanoplastics. And what we see, similar to what we have seen for particles, is that they actually end up in many of the organs and biological systems. What we also can see is that, and Hubert was completely right, that it's not easy to draw conclusions on exposure to date from those studies. And that has largely to do with the pie chart on the right side here, when we actually summarize the different assays that have been used to detect those particles. You can see that there's a vast array of different methods with spectroscopy, Raman, and FTR. It's basically the main analytical approach till the end of 2023, but also with pyrolysis, GCMS, and others being there. And that data is not necessarily easily combinable. The other aspect that really limits us to date is that although we have many studies now, or at least 50 plus studies, if we look at the sample size of those studies, we have to admit that they are in the very small N range, meaning that many of the studies are less than 20 people, and basically very few are between 20 and 50 people, and very few are basically over 100. So in all honesty, our knowledge about basically the spread of micro nanoplastic exposure into the population, as well as basically the demographics of that, are largely unknown. We also talk about MNP, and on a slide the word MNP looks very simple, but in actuality micro nanoplastics is a hugely complex exposure. That is not only true from basically the morphological aspects of micro nanoplastics, thinking about size fractions, which Hubert already talked about, micro and nanoplastics, but also the morphology. Are we talking about fibers, fragments, films, foams, filaments, flakes, particles? They all change, they all have basically different properties. But it becomes even more complex when we actually think about the chemical composition, and this is basically on the right side, a picture adopted from the Weifman paper in Science Advances in 2018 already, basically listing that there are over 10,000 Cas numbers associated basically with plastics, may that be the monomers, may that be the additives or processing aids that are actually used within the production of micro-nanoplastics. So we're talking about an exposure that is chemically very, very diverse and complex. That also leads to basically different physical chemical properties, thinking about solubility, surface chemistry, but also Hubert also pointed out, we also have to think about the absorbed chemicals, may that be microbes and biofilms, chemicals or metals, because micro-nanoplastics in itself are basically great carriers of other exposures. So this is really what we are confronted with if we think about what micro-nanoplastics is. So in this background, we started the AURORA project. The AURORA project is a 5-year project, so it's the only project within the CUSP cluster that continues 1 year extra. So we're now basically at the end of year 3 of a 5-year project. So we still have 2 years to go, but as you will see, we also have still quite a bit of work to be done within this project. So what is really the premises of the AURORA project is that we wanted to understand what is the exposure to micro-nanoplastics to pregnant mothers, how does basically then the micro-nanoplastics affect placenta health, either directly or indirectly also passing micro-nanoplastics to the fetus, and what does it actually mean to early life health. So we're actually trying to follow the exposure basically from pregnancy to placental transfer to exposure to the fetus to the outcomes in early life health. So in the next slides, I will basically try to go through some of the logic of the project. The first question to be answered is, why do we actually focus on early life health? And basically there are 3 very major points here. As we know from particle toxicity, but also from EDCs and other chemicals, early life health and the pregnancy is really a very vulnerable time period. It's also true that when we look at basically exposure to the fetus side, effects that actually happen in early life health basically have a large effect also to adulthood. So from a public health standpoint, it's very important to actually understand these health effects early in life. The other third one is that when we go back to what we already know on chemicals and particles, there is good previous evidence of placental transfer of particles and chemicals, which also are related to impaired child development. So there's reason to believe that particles in itself and chemicals that we also see in micro-nanoplastics do have an effect on early life health. The major question, however, is, do micro-nanoplastics do something differently than we actually already know when we look at particle toxicity or when we actually look at these chemicals' risk in itself? So what we did, and we are very happy that this is now being published very, very soon in the Journal of Microplastics and Nanoplastics, the paper by Emily Christopher of the IOM in Edinburgh, Scotland, where we basically took the risk assessment frameworks that currently exist, may that be particle toxicity or may that be chemical risk assessment, and mapped that back to what does that mean for human health risk assessments for MNPs and early life health. And what I display here are the 15 gaps that we identified that are there that actually precludes us at this moment to do a human health risk assessment of MNPs. What those are basically summarized in 3 categories. The first one is basically issues in exposure assessment. We actually have a dearth of information about what the exposure of the population is, and that has to do with characterizing that exposure. It has to do with exposure duration. What do we know about bioaccumulation? What do we know about excretion rates? These are all still questions that we have to answer. The other gaps that are identified have to do with the hazard assessment. And here we have to think that we have to do polymer hazard assessment. We have to think about what do these particles do, but we also have to integrate the chemical hazards that are associated. And these are, of course, not unique things. They're actually combined in these MNPs. And that actually means that we cannot go at this moment to hazard values, which also basically means that it impedes semi-quantitative risk assessment approaches, or let alone quantitative risk assessment approaches. So these are really large gaps that we still have to close. There are also gaps in what I would call paradigms and resources, which has to do with, okay, but if we actually want to test these large array of different micro nanoplastics, how can we do that actually with non-animal testing? Because we are having here hundreds to thousands of micro nanoplastics, which we cannot do in animal experimentation, or I think that's not what we want as a society. We have to think about where do we get the reference materials, what do we do with the existing paradigms that we have in risk assessment, how do we actually standardize reporting of these new exposures. These are all questions and gaps that we have to answer. So in AURORA, we basically start to try to solve some of these gaps. And the first one is basically how do we actually do micro and nanoplastic quantification in complex tissues. So here we are actually taking the leap to focus on human materials and see how we actually can do that quantification. As Hubert already indicated, you basically have two flavors of this, and I know that it's a little bit of a dichotomy that I put here, but you can think about this about spectro and microscopic techniques, which basically look at particles. Those can give us basically very deep phenotyping, but as I will show you in a minute, are relatively slow. The other way that we can go is basically using chromatography or mass spectrometry, in which we can get mass-based assessments of particles and chemical constituents of particles, but we actually cannot get the morphology from the chromatography and mass spectrometry. So these two techniques really have to go hand in hand if we actually want to solve this issue. So on the MNP assessment in AURORA, which I call this now slow but deep, we're actually using a wide variety of different spectroscopic and microscopic techniques. The reason to do that is that Raman FTR are good techniques to look especially at microplastics, to actually get into the nano range, asks for more sensitive techniques, and that's what we are working on, because we are especially interested also in being able to measure these nanoplastics with microscopy and spectroscopy. We actually have come quite an end, I would say, at this moment, and this is really work of Laura Zoutendijk, a PhD student of Florian Meyer at Utrecht University within the chemical department, which also produces for us reference materials. So we actually are producing our own nanomaterials with embedded fluorescence labels, so actually can use those in experimentation. So what you see here, and sorry for showing perhaps for some more graphical pictures of placenta material, but what we basically have developed is techniques to take this very complex matrix of placentas, are able to digest those placenta materials into a neat fluid, filtrate those biological samples, and then get that on a filter or a gold filter to do basically spectroscopy. And the result is actually shown here on the bottom right side, and this is actually showing where we are spiking our samples with our fluorescence particles. In this case, these are 50 nanometers, so really particles in the nano range that are being added to the sample, and we actually are able to recover 90% of those particles without actually having a change also in morphologies of these particles. So we are quite confident at this moment that we actually have developed techniques to be able to measure these particles in biological samples. To show you that this also can work in real-world samples, although in an experiment, and this is again work from Laura Zoutendijk and Britt Juffer, and together with Marcus Vrijberg in Finland, where we actually took placentas. So this is a whole placenta donated after birth, quickly after birth, that is actually put in a system where we actually can perfuse the placenta with artificial blood. And what we do, we actually spike the artificial blood with micro nanoplastics to study how the micro nanoplastics, when we perfuse a whole placenta, how they actually distribute from the maternal side to the fetal side, and where do these particles end up. What is shown here on the right side, I think, is very exciting research that we just haven't gotten in the last month. Is actually showing a slice of the placenta where the actin is labeled in red and where the polystyrene is labeled in green. And what you can see is actually that we are able to actually trace the micro nanoplastics within the structures of the placenta, and actually are now able to see where do they go, where do they accumulate into the placenta. So this is actually showing that the techniques that are being developed are suitable to do this experimentation. However, although this is usefully advanced work, it does have a limitation, and that limitation is basically scalability and complexity. The scalability issue is that these techniques we can use on a few samples, but as I try to basically indicate, is that we need actually larger sample sets to actually get to the exposure assessment. The other thing that we actually have to realize is that we are not talking only about micro nanoplastics, but as laid out before, we are talking about additives, we are talking about transformation products, and co-transported environmental pollutants. So in a paper by Cornwell Shao of Emory University in the US, from the lab of Doc Walker, we basically have now a paper in press in the Annual Review of Pharmacology and Toxicology, where we lay out that we actually need an exposomics framework embracing that complexity to actually understand the effects of micro nanoplastics. So what we are doing at the moment is to do what I would call fast and wide exposure assessment of micro nanoplastics. And this is really pioneering work by Doc Walker of Emory University, where he has been able to build a high throughput workflow which is called FastME, where we are linking pyrolysis, HRMS, which basically Hubert showed and which most of us have used until now, and that basically gives us the polymers and monomers. But that's actually directly coupled to alkaline assisted hydrolysis, liquid chromatography, HRMS. And what that actually enables us is to get the bounded chemicals within the monomers. And so that is actually where we get into these chemicals that are there. We have coupled that to a third high resolution mass spec technique, where we actually are measuring, which is the untargeted liquid chromatography, where we actually measure the free chemicals, may that be flame retardants or phthalates or others, and at the same time can measure the biological readout. And so we are actually intersecting the signal of these free mass specs to actually come up with not only the monomers and polymers, but also the associated chemicals and the free chemicals to really embrace that complexity of what really MNP exposure is. This is just showing some preliminary results. This is showing basically the results of polyethylene, polystyrene, PVC, and so on, where we analyze placenta and blank samples. And what you can see is that we see plastics within the placenta material, but basically at a level that is about 100-fold higher than backgrounds. So we actually have sensitive methods enough to be able to detect these monomers and polymers within the placenta. This is actually showing the first results of the alkaline assisted hydrolysis method. And what you can see here is exactly what also was in the earlier paper that I showed, is that on the y-axis here you have the unique feature count. So in all means and simplicity, let's just assume that these are all unique chemicals. And then you can see that by actually running this, we have about 1,000 to 2,000 associated chemicals that come with these monomers and polymers. So again, emphasizing that this is really a complex exposure and that we need these different techniques to really get the full picture. So next to actually doing this work on better exposure assessment, we basically identified these gaps in hazard assessment, in vitro assessment, and that's where we are developing a tiered approach where the first tier is basically what we call screening and prioritization. Then it becomes more advanced in basically still in vitro by having more complex co-cultures. Then we can take that one step further and do placental perfusion experiments and take that one step further again and do developmental toxicity studies, which basically we can do with zebrafish or stem cell test systems. As I said, we do that with testing monolayers, co-cultures, multilayers, and organ-like. What we have done today is basically these monolayers and co-cultures with basically BWO cells, which are basically placenta cells, UVAC, which are basically umbilical cord, cell lines, and H295R, which basically is adeno gland cells, which basically reflect more the stereogenesis systems, which of course is important when we think about EDCs. In co-cultures, we actually put those cells together or build multi-layer test systems. I'm not going to go into all the details, but I think it's important to share these first results that we have. And so this is basically a very heuristic summary of the tier 1 and tier 2 summary, in which we actually to date have tested 25 polymers, different polymers, different sizes, also into the nano range. And when we actually look at tier 1 and tier 2, and we actually look at cytotoxicity, membrane integrity, oxidative stress, gene expression, cytokine secretion, Hubert already pointed to IL-6, IL-8, IL-1 beta. When we all measure those, I think what we actually conclude to date is actually we see relatively minor effects with these pristine plastic particles that of course are more simple than what we actually see in real life. And if we see effects, it's basically at very high concentrations, which are not biologically relevant when we think about human exposure. At least that's what we think at this time. So that leads to a discussion, basically, are we looking at the right endpoints? And so we're actually doing deeper phenotyping now to see basically if we have missed certain things in the biological system that are important. The other one is basically that we actually do have to look at more polymers and real life samples to be able to look further into this. The good news, of course, is that we have been able to actually build this tiered system in which we actually can prioritize the different testings of micro nanoplastics. The real leap that we are taking in AURORA, although the other ones are significant leaps as well, this is probably the furthest leap that we actually are taking. And that is actually that we currently are analyzing over 800 placenta and cord blood samples. So this is actually much larger than studies to date, which I think is really necessary, which we actually have obtained from ongoing prospective birth cohorts, BISC in Spain, led by IOS Global, and the EnvironAge birth cohort led by the University of Hasselt, which basically provides both the placenta samples and the cord blood samples. We analyze those samples by our mass spectrometry and then basically do confirmative analysis by spectroscopy to also look at the morphology. We link those exposures to placental function, think about placental blood flow and other measures of placental function. We then link that to what we call system homeostasis. So what is actually perturbated within the human system is that immune inflammatory responses, oxidative stress, accelerated aging, endocrine function, or more a system biology approach by actually doing metabolomics, so very wide screen of biological effects. Given that these are birth cohorts in which the children are now actually at the max of the age of 12, we actually can also look at fetal growth and child health outcomes to see basically what are the long-term chronic effects, which Hubert also identified as one of the largest gaps that we have to date. So this is exciting work, and we expect to have all the exposure data in within 2024 to really in 2025 make these links to all the biological and health outcome data. As Hubert pointed out, we are using basically historically collected materials, which there is always a risk for contamination, and we are fully aware of that. So we also have done a de novo collection of 25 samples in which we actually have collected everything in glass tubes and aluminum foil and so on, to actually see if the contamination issue is going to hamper our interpretation. I think the results until that now is that, of course, there is a chance for contamination. We do feel that that's not going to hamper our interpretation, because it, in essence, is going to be a stochastic background exposure that we are going to see. So to go back to what we laid out in this roadmap, the Human Health Risk Assessment Framework for MNPs and Early Life Health, I basically put some squares around the boxes where I think AURORA at this moment is really contributing on getting some steps into understanding what is going on. By far, we are not going to come with definitive answer. This is really the first set of studies that delve into this area. And there are large gaps that I really think we actually need to address altogether. This field is too large and too complex to only do that mono-institutional. So all resources within AURORA are open, and please get in contact with us to really set up joint research endeavors to really move this forward. Just wanted to share some of the publications that have come out. These had a lot to do with which techniques can be used. The paper in the Science of the Total Environment was the first one where Hanna Dusa of Utrecht University showed basically the uptake and transport and toxicity of MNPs in the placenta. The paper of Xiao has actually shown basically what we need to do. And then we basically have our roadmap, which I really encourage everybody to read and reflect on, and basically give feedback to us on what we need to do. Of course, I have to thank the European Committee for the funds, but also to thank the CUSP colleagues for all the sharing of resources and information, which is really essential to move this field forward. But also all the people within the project, which are all listed here on this picture. And with that, I just wanted to conclude and thank everybody for their attention, and I'm looking forward to question and discussion.
Okay, thank you very much, Roel. I think an excellent and very rich presentation. I was particularly interested on the cohort study and the groundbreaking stuff you're trying to do with that. Now, we do have quite a few questions, so we are not going to be able to get to all of them. We may come back to answer some of them written-wise. I don't want to commit to anything, but thank you, everybody, for all of the questions, and I will just stay on another maybe 5 minutes, if that would be okay, to answer a few of the questions that I had. And I'm just looking here. I had, so one of them was, let me just see, a quicker question about the study. Well, here's one. Have measurements of MNPs across multiple specimens, example, blood, urine, feces, collected from the same subject, been performed? And if so, how strongly do the measurements across specimens correlate? I don't know if either one of you, that's one of them, and I have a few others.
I don't recollect all the papers. Many of them have been mono until now. Mono is basically sample. What we do in AURORA is actually collecting both blood from the mother in these pristine collections of basically the placenta of the fetus and urine and feces, to actually see how the pharmacokinetics work. And so I think these are the studies that are really needed to see how actually the kinetics work. And I'm just looking at Hubert if there's any addition to that.
No, I'm also not aware of that rule. And the problem is, yeah, of course, we are measuring a surrogate. You know, we are measuring the polymers, the mass of polymers. It's very difficult to do in biological tissues, really look for micro and nanoplastics with microscopy. But I'm not aware that the study with several media. It would be interesting, but quite cumbersome to do. Yeah, you know, you cannot dose people with an amount of micro nanoplastics. You know, that would be ethically not acceptable. So it's an interesting study, but I'm not aware of it, that it has been performed.
Okay, thank you both. So here's a question around the football study. Are the levels of 6 ppd being analyzed in the football study?
In biological tissues?
Yeah, I think so.
No, we don't have that method yet, but we are working on it, and maybe yeah, we can look at it in the urine samples or in the blood samples. We have it on our wish list to our analytical group in the institute, so hopefully we have a method soon. Yeah, but we have not done it yet.
Okay, thank you. How much evidence is there for microplastics deposition in the human brain? Would they be expected to accumulate in the brain?
I think at this moment that would all be speculation at this moment, because I think we are really at the cusp of being able to do that. But as I pointed out, in principles, small particles, as we know also from air pollution, nanoparticles do end up in the brain. So I would say that it is a reasonable conjecture to actually say that micro-nanoplastics would end up there as well if they are in the right size range. It's more basically our analytical techniques at this moment good enough to actually measure those and trace those well enough, and are they sensitive enough? And if they are, I'm pretty sure that you should be able to detect.
Do you want to add anything to that?
No, I also think we know for air pollution, like Roel said, you know, that if you inhale it via the nose, that it will end up in the brain, because there is and there's no reason to think that micro nanoplastics are not behaving in the same way. But yeah, I don't think that it has been studied yet. And it's the methods, the analytical methods, are they sensitive enough to detect?
Okay, here's a question in terms of risk assessment, to go this way. What do we know about the reference doses, the RFDs, or any alternative recommended safety levels for MNPs?
I don't think that has been. It's not possible. We lack a lot of data. You know, on acute effects, like also Roel presented, I think we have not seen a lot of dramatic changes in acute effects, but in the chronic effects, it's the jury is still out there, and then we are really talking about long-term, very long-term effects, like asbestos. I keep referring to that. It took 25 years before we discovered that there is a problem with asbestos exposure and specific tumors in the lung. So it at this moment we cannot say anything about this.
Okay, thank you. Another question, are there efforts being conducted to standardize both the sampling and analytical methods to enhance the accuracy of testing results on MNPs in human organs and fluids?
Yes, and this is actually a working group within CUSP that actually is standardizing reference materials, is trying to standardize basically the way that we collect and test, and standardizing the way that we actually report. These are all very important things if we actually want to be able to link evidence bases together from different labs and different studies in the end. So this work is ongoing, but at the same end, of course, this is very much still pioneering work. So we're not talking about methods that basically have established themselves to the level that we actually can talk about standards. So I think that's still where we are at this moment.
Okay, thank you. How about the stiffness of MNPs as potential contributor to their translocation into the human body?
Well, yeah, they are particles, and they are, you know, in the study from Italy, in which they looked at the particles in the aorta, I assume that they are stiff enough to be present as particles in the body. But yeah, it's difficult to detect. It's really looking for a needle in a haystack, you know, if you want to look for particles in biological tissues. Unless you mark them or with a fluorescent group, for example, then it's easier. But it's not trivial to do this electron microscopy. You have to collect a lot of samples and study a lot of samples in order to say something about that. But yeah, it would be interesting information to have, absolutely. But we, this moment, we don't have it.
Okay, thank you. Well, I'm going to I'm going to kind of wrap this up because I know we've gone a bit over. I think we could probably spend another hour or two answering all the questions. There's so much, and we have these two great experts here. A lot of people have been just saying what awesome, what excellent talk, and lots of information, lots more questions about some of the sources and ways you're doing this research. So I just want to thank both of you again for this great intervention, and the slides that will be made available so people can use that to follow up, and we'll be taking a list of all the questions and see how maybe we can get back to some of them. So thank you very much, Paul and Hubert, and I will hand over the floor to Ale.
Great. Thank you so much, Shannon, and thank you, Hubert and Roel, for your excellent presentation and for this important research. And thanks also for the great comments and questions from the participants. I'll wrap up now with some quick announcements. There will be a video recording available on CHE's website soon, and you will all receive an email with a link to the video. And also, please join us for the following upcoming webinar, for which the information is also being provided in the chat. So on Tuesday, July 2nd, CHE Alaska will be hosting Mapping Environmental Injustice, Disparities in Chemical Exposures, and Neurodevelopmental Outcomes. We encourage you to support the work of any and all of the groups involved in the EDC Strategies Partnership. You'll find links to each group on CHE's partnerships page. If you appreciate these webinars, please consider a tax-deductible donation to CHE and/or any of these partners. And with that, we'd like to thank all of you again for joining us today. Thanks again to Hubert and Roel for taking the time with us to talk with us about this important work, and to all of our EDC Strategies partners, and to Rachel Massey for her help behind the scenes. Thanks, everyone.